Long-term potentiation has been reliably demonstrated in the hippocampal formation in the laboratory. Scientists present a rapid series of electrical impulses through a stimulating electrode and record the responses in hippocampal neurons with a second recording electrode. The dentate gyrus region of the hippocampal formation receives neural input from an area known as the perforant pathway in the hippocampus. An electrical pulse is delivered by a stimulating electrode placed in the perforant path. A recording electrode located in the dentate gyrus records the size and duration of the resulting EPSP. The resulting EPSPs can be measured. A single threshold pulse of electrical stimulation results in a typical EPSP. However, a rapid succession of intense stimulation induces LTP. Following the intense stimulation, the cells in the dentate gyrus will respond to subsequent single pulses of stimulation in the same pathway with larger and longer-lasting or potentiated EPSPs. Scientists think that such changes in the intensity and duration of PSPs may be a potential mechanism for strengthening synaptic connections that underlie learning. Repeated pairing of stimuli presented close together in time, such as a telephone number and a person's name, may induce LTP-like changes in neurons and subsequently strengthen the neural connections that allow us to associate the phone number 555-6789 with our friend Joe. Laboratory research on hippocampal cells has revealed that specific patterns of neural activity, such as intensive stimulation or concurrent activity in pre- and postsynaptic neurons, changes the time course and intensity of postsynaptic potentials, PSPs. Intense or very rapid stimulation of neural circuits in the hippocampal formation leads to long-term increases in the size of the excitatory postsynaptic potentials, EPSPs, in the postsynaptic neurons. This increase in the size of the EPSPs is long-term potentiation, or LTP. Let's explore what changes occur at the neuron cellular level that make long-term potentiation possible. The postsynaptic dendritic spines of neurons in the CA1 region of the hippocampus contain two kinds of receptors for the neurotransmitter glutamate, NMDA receptors and AMPA receptors. The NMDA type of glutamate receptor is essential for some forms of LTP. The NMDA receptor subtype is permeable to calcium, Ca++, but the channel is blocked by magnesium, Mg++. That is, the NMDA receptor channel is normally blocked, so even if glutamate were present, it could not activate the NMDA channel. It would activate only the AMPA channel. The AMPA receptor controls a sodium channel. Glutamate opens the sodium channel, resulting in the influx of sodium into the postsynaptic dendrite. The influx of sodium results in the generation of EPSPs, or depolarization in the postsynaptic cell. This depolarization, due to the influx of sodium into the channel controlled by the AMPA receptor, results in the ejecting of magnesium from the NMDA receptor. The ejection of magnesium from the NMDA receptor allows calcium ions to flow into the dendrite, resulting in a cascade of second messenger-related events involving calcium-dependent enzymes, which ultimately results in the insertion of more AMPA receptors into the dendrite. As a result of LTP, the synapse has now been strengthened. This synapse will respond more strongly to future releases of glutamate with larger and longer-lasting EPSPs, which means you have learned the association or connection between Joe and his phone number.